Next, we are going to see about the pollen grains. It was the male gamete of plant. So, now structure of pollen grain. Pollen grains are derived from the microspore. Yesterday class we have seen that. And then pollen protoplast, it is like a cell. So pollen protoplast, that is pollen cytoplasm, has a centrally there is a nucleus also present. The wall into two layers, that is inner layer is called intine and outer layer is called exine. Intine is generally thin and uniform in thickenings. They are made up of pectin, hemicellulose, cellulose and callose. Exine is generally thick and it is made up of cellulose, sporopollenin and pollen kit. So exine is not uniform, thin at certain areas, thick at certain areas. Most of the pollen grains are ornamental and it is associated with the germination of pollen grains. So sporopollenin is absent in germ pores alone. So pollen grain contains some pores, so that is called germ pore. In that region sporopollenin is absent. So here you can see the structure of pollen grain. So ornamental exine is there, it was thick, and then thin smooth membrane of intine is present. Inside that cytoplasm and nucleus is present, and it has a germ pore. So through that it has to grow and inside the stigma, so it has germ pore. And then next is sporopollenin. Sporopollenin is found in the exine of the pollen grain. The wall material of sporopollenin contributed by both pollen cytoplasm and tapetum. So it is derived from carotenoids present in the plants. Carotenoids means colored pigments present in the plants. So why it has to be present in the exine means it has a strong resistance to physical and biological decomposition. So it is protective in function. Inside that nucleus and cytoplasm is important. It is the future gamete of the plant. So it has to be protected. So it is resistant to physical and biological decomposition that was maintained by sporopollenin. So that the exine contains sporopollenin. And it can withstand in high temperature and strong acid and alkali and enzyme action. So any structure cannot destroy the sporopollenin. So mainly it is responsible for the protection of pollen grain. So through the sporopollenin we can preserve the pollen for long periods in fossil deposit and also during its journey, because all the pollen has to transfer from male pod to female flower. It has a strong journey. So during the journey of the traveling of the pollens, it was protected through sporopollenin. Then pollen kit. It also contributed by tapetum and colored yellow-orange pigments, that is carotenoids or flavonoids. And it has an oily layer of thick viscous coating over pollen surface. So why it was present means it is attracted by the insect and protect the damage from UV radiation from the sunlight. So this also present in the exine. It is an oily layer forming around the pollen surface. It is used to attract the insects and protect the pollen from the damage of UV radiation.